By this time, we know that genetic material, whether in prokaryotes or in eukaryote, is made up of chromosome. And this chromosome is made up of proteins and nucleic acid. There are two types of nucleic acid, DNA and RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So let us study the structure of DNA. As far as history of DNA is concerned, in 1869, Frederick Michel for the first time extracted it and he gave it the name nuclein. Then after a very long period, in 1953, Maurice Wilkin and Rosalind Franklin, both these scientists, they gave the X-ray diffraction of DNA, and on the basis of this, James Watson and Francis Crick, they gave the double helix structure of DNA and they got the Nobel Prize for this structure also. DNA, this is the largest polymer of the body. And we all know that what exactly is a polymer. A polymer is a compound that is made up of many monomers. So polysaccharide, protein, lipids, DNA, RNA, these are the polymers in our body, and out of all these 5, DNA is the largest polymer in the body. Length of DNA. What do you mean by the length of DNA? Length means the number of base pair. Base pair means nucleotide. Because DNA is a polymer of nucleotide. As far as prokaryotes are concerned, in E. coli, the number of base pair are 4.6 into 10 raised to the power 6. And in eukaryote or in human specifically, the number of base pair 3.3 into 10 raised to the power 9 in N condition and 6.6 into 10 raised to the power 9 base pair in 2N condition. So all these number of base pair when multiplied by the distance between two nucleotides, they give us the length of the DNA. Now, just now I said that DNA is a polymer. So now let us check the monomers that make the DNA structure. Monomer of DNA. Number 1. It is made up of ribose sugar. Ribose, this is a pentose sugar having 5 carbon in this. This is a pentose sugar. And this sugar is a ribose sugar. This is ribose sugar. But I am telling you that DNA is having deoxyribose sugar. So in DNA, this oxygen is not there. It is not there. So instead of this, it is having simple H here. So this becomes deoxyribose sugar. So whether it is ribose or deoxyribose, both are pentose sugar. So this is the ribose sugar. And when we have to number it, we are going to number it like this. 1, carbon 2, carbon 3, carbon 4, and this is carbon 5. The second component of this is phosphate group. Phosphate group is PO4. This is common in both RNA and DNA. And third is nitrogenous base. There are two types of nitrogenous base. One is called purine and another is called pyrimidine. Now, what are nitrogenous base? Nitrogenous base, these are heterocyclic ring. There are 2 rings in purine. Purine is having always 2 rings. Their structure is like this. And pyrimidine, they are made up of just 1 heterocyclic ring having nitrogen compounds in it. Okay, so these purine or pyrimidine both are made up of heterocyclic ring of the nitrogenous compound. Now purine, it is having, or again this is of 2 types, adenine and guanine. And pyrimidine, these are of 3 types, thymine, cytosine, and uracil. But because we are doing the structure of DNA, uracil is not found in DNA. Uracil is found only in RNA. So we will not write this here. Right? So these are the 3 components of DNA. Now let us see how these components, they are attached to each other by different bondings. Now let us study the types of bonds. The first bond that is present in the DNA is glycosidic linkage. Glycosidic linkage, this is the linkage between nitrogen base and the sugar, pentose sugar. So if this is the pentose sugar, they are attached the nitrogenous base. This may be A, T, U, C, N. The second one is phosphodiester linkage. This phosphodiester linkage is between the pentose sugar. This is the pentose sugar. I am drawing 2 pentose sugar here, like this. This is 1, 2, 3, 4, 5. These are the carbon number 1, 2, 3, 4, 5. Now this phosphate group is present between this carbon 3 and this carbon 5. Alright. So between carbon 3 and carbon 5, this phosphate is attached, and because this linkage and this linkage, these are ester linkage. So we are calling it phospho because phosphorus group is there, phosphodiester linkage. The third type of linkage is hydrogen bonds. Hydrogen bonds, these are between nitrogenous bases. Say, this is the pentose sugar and there is A, and this is another pentose sugar, and here it is T. So in between these two, there is hydrogen bonding. Nitrogenous base, they always combine to each other by means of the hydrogen bonding. So this is clear that there are three types of bonding here. Glycosidic linkage is between the deoxyribose sugar and the nitrogenous base. Phosphodiester linkage, this is the linkage which is present between two deoxyribose sugar and between the phosphate group. And the third is the hydrogen bonding, that is between the two nitrogenous base. Now let us study the chemical structure of a part of DNA. Now, first of all, I would suggest that draw 4-deoxyribose like this. Then it would be easier for you. Right? And you can draw one more here. Similarly, draw 4 bases there, but now in the reverse direction like this. You have to draw these parallel to these. The third one and the fourth one. Okay, so we have drawn here deoxyribose sugar. Now we will draw the linkage, phosphodiester linkage between carbon 3 and carbon 5. 1, 3, carbon 5. 3, carbon 5. Similarly, draw these here also. 5, 3. 5, 3. 5 and 3. So, we have shown here the phosphodiester linkage. Right? Now we have to attach the nucleotide base to this backbone. Now let us attach these. Say it is adenine here and it is thymine here. So they will combine by the double bond. It is cytosine here. So cytosine, it always combined by triple bond to guanine. Say thymine here, again this will combine by double bond to adenine. Similarly, you can draw any other base, guanine, combining to cytosine like this. So this is a part of the DNA. Now look at this structure. Again concentrate on the numbering of carbon atom. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Similarly, 1, 2, 3, 4, 5. Now look at here. Now in this angle, at the lowermost one, this one, here carbon number 3 is free. So this is called 3-N. On the other hand, at this end, you can see carbon number 5 is free. So this is called 5-N. Similarly, look at this strand here. Carbon number 3. This is carbon number 3 here. This is 4 and this is 5. 1, 2, 3, 4, 5. Similarly here 1, 2, 3, 4 and this is 5. So here carbon 3 is 5. So this is 3-N. And similarly at the last here the carbon 5 is free. So this is called 5-N. So this strand is running in 5- to 3- direction and this strand is in 5- to 3' direction. This we say that these are anti-parallel to each other. When we say that both strands of DNA, they are anti-parallel to each other, it means one is running in 5' to 3' direction and the other one is running in 3' to 5' direction. Now let us draw double helix structure of DNA. We have to draw the strand of DNA like this. Strand have to be like this. You need not to draw it like this. This is the wrong structure because it is having minor groove and these major groove. So that is why your structure has to be like this and not like this. This is the wrong way of drawing DNA structure. Now in this, we are showing the nitrogenous base. These are the nitrogenous base here. One complete turn of DNA is this much. So one complete this, one complete this. This is the breadth of DNA, that is 20 angstrom. The total length is 34 angstrom and the distance between two base pair is 3.4 angstrom. 3.4 angstrom means 3.4 into 10 raised to the power minus 10 meter. This is known as backbone of the DNA, and we have done that this backbone is made up of phosphate group and deoxyribose. And the nuclear bases are projected inward. Adenine plus guanine, that is total number of purine bases, they are always equal to total number of pyrimidine bases. And it is because of this ratio that the distance between the two strands is maintained equally throughout the long strand of the DNA. So according to Watson and Crick structure, there are two strands. These two strands are anti-parallel. Anti-parallel, I have informed you, one is in 5' to 3' and the second one is 3' to 5'. And when we are saying 5' to 3' or 3' to 5', it means the position of the carbon that is not linked to the phosphate group. Then the third one is the same, that is also called Sharga fluid. That this ratio A plus G is equal to T plus C. This is maintained. Then another one that I have just informed you, distance between two base pair is 3.4 angstrom and total length of one strand, one complete round, one complete turn you can say is 34 angstrom, breadth is 20 angstrom.